Now I would like to introduce our webinar speaker, Dr. Patrizio Giacomini. Dr. Giacomini graduated in 1980 at the University La Sapienza in Rome and obtained a specialty school degree in infectious diseases in 1985. His main research interests are HLA, tumor immunology, and both natural and recombinant antibodies. Recently, much of his activity has been focused on nanoimmunotherapy. He has also a keen interest in the methods and social implications of biotechnology. Dr. Giacomini worked at several international institutions, including Scripps Clinic and Research Foundation and Columbia University. Over the past 30 years, he has taught immunology at several Italian universities and was appointed to serve in the staff of university biotechnology centers. Presently, Dr. Giacomini is group leader at the National Cancer Institute and has active ongoing collaborations with the academia in the pharmaceutical industry. He is author of over 100 publications and peer-reviewed journals. His research has been supported by private companies and investors, as well as public funding bodies in Italy and Europe. So, the title of Dr. Giacomini's presentation will be Antibody Epitopes and Protein Conformation. So, Dr. Giacomini, I'm handing, as one would say, the cursor over to you. Please take it away.
Well, thank you very much for your nice introduction. And I would like to start by saying good afternoon and good morning to all of you. And thanks for the invitation. It is really a pleasure for me to have the opportunity to talk about a topic I like very much. And as you can read in the title, it's antibody epitopes and protein conformation. Well, of course, I also thank all of you. I see that you are numerous, you are attending the seminar, and thank you for joining us. I work at the Italian National Cancer Institute, and I am an immunologist. I think that we can organize this way. At the end of the webinar, I will be happy to take questions. But in case you think about what I'm going to tell you, and you have additional questions and you have not asked by the end of the seminar, please feel free, write down my email address that you can see in the slide, and then you feel free to just email it. What I'm going to talk about today is largely based on a study of ours that has been just published in the European Journal of Immunology, so you will find there many more details. So now I will give you a brief introduction, and then we rapidly switch to the hard data that is the reason why we are all here. Okay, the first statement I'd like to make is a very simple one, and one that we all agree upon. Epitope mapping is when you know exactly where your antibody binds to your protein of interest. Let's make just two basic examples. There are essentially two reasons why you want to map an antibody epitope. As you can see here on the left side, the first reason is when you have many antibodies, like in patient sera, but you do not know which antigens and which epitopes they bind. In the second case, reason 2, it is somewhat a simpler case, is when you have already selected a single antibody. For instance, you have developed a murine monoclonal, therefore you know the antigen, but you actually do not know where the epitope is located on. In this right panel, you see that there are 3 different epitopes, but only the antibody binds only to the blue triangle, only to one of the 3 epitopes. Now, whether you want to develop a sensitive serum profiling assay, that is reason 1, or you want to validate a potentially diagnostic or therapeutic antibody, and this is reason 2, whichever the case, knowing the epitope, learning the position of the epitope will give you a lot of information. Without the epitope, you cannot be sure that the patient is responding to disease, and you have no clues, that you do not actually know whether or not the antibody that you have selected is a good candidate for biological development, for biotech development, to make a therapeutic antibody, for instance. So to know the epitope is the first thing first point I want to make, is to know the epitope is very important. Okay, so these are just two basic cases, but if you have a glimpse at the PEPperPRINT website, you will see plenty of examples. Whatever the specific reason, reason 1, reason 2, reason 3, whatever the reason, today I will focus on a very specific issue that is often neglected, in my opinion. An issue that epitope mapping may help to alleviate and possibly resolve. So actually, I will take a few minutes of your time to further elaborate on case 2, on reason 2. I will try to warn you about some possible risks that are related to the use of antibodies when the epitopes are unknown or poorly characterized. I will focus on human leukocyte antigens, HLA class 1 molecules. In the lab, we use murine monoclonals to probe the conformation of these molecules. And my purpose, what I'm going to talk today, is this is the aim, the general aim of what we do in the lab, is to determine whether certain regions of the HLA molecules bind specific antibodies, and whether the epitopes that are carried by these regions are conformational or linear. So let me just start giving you some background about class 1 MHC antigens. These antigen-presenting molecules are heterotrimers, cell surface heterotrimers. They are made by 3 non-covalently associated subunits: a polymorphic heavy chain that is depicted in yellow, a non-polymorphic light chain, beta-2 microglobulin in blue, and a small peptide antigen that is in red. The antigen fits the so-called antigen binding groove that is formed by the heavy chain, and it's kind of a chair formed by 2 alpha helices here, and a bottom, the bottom of the antigen binding, as I'm saying. These are the regions where murine monoclonals are most often developed. They are highly immunogenic. Now, are they folded? Are they unfolded? So this is the question. This question is complicated by one important aspect. As you certainly know, these antigens are highly polymorphic. There are more than 6,000 different alleles that encode highly homologous heavy chains, and these differ from one another sometimes by just a few amino acids. So polymorphism is a major technical challenge with these molecules and with other molecules whenever you deal with related families of proteins. So an additional question I want to ask about the epitopes that are carried by this structure is whether the epitope is carried by allele 1, 2, 3, for instance, from the B locus, and not by alleles 6, 7, 8, and so forth. So an antibody of restricted specificity is, of course, very important because there are subtle differences, also functional differences, among the products of the different loci and the different alleles. So I want to be able to tell whether my antibody binds A and not E, or C and not G, and so forth. So the major problem to address this issue is that conformation and polymorphism combine within the same molecule. So if I want to know where is the epitope, and I want to make sure that the epitope is not shared by all these molecules, in principle, I will have to test the more than 6,000 alleles in different conformations. And I can hardly think of a simple method by conventional protein technology whereby this can be accomplished. This is not an academical issue. To have an antibody that binds a known epitope carried by a well-folded protein and not shared by the other alleles means that you can use this antibody to predict, to make diagnosis, to establish a prognosis, and in general, there are important therapeutical consequences. So when we asked advice from Dr. Volker Stedler from PEPperPRINT, our goal was to map the epitope of a murine monoclonal antibody to class 1 HLA. This antibody had been used by several groups in recent years. So we prepared a glass chip that displays hundreds of overlapping class 1 HLA peptides. And we realized almost immediately that one single epitope stands out. In spite, on the chip, there are thousands of different spots. It is interesting to note that this antibody that we selected for epitope mapping binds strongly and has almost zero background, as you can see both here and in the graph. And in addition, 3 different types of overlapping peptides identify the same epitope. So it is not dependent on the length of peptides. So 400 times the background. And the signal is clean, and unrelated peptides or peptides located in other regions of the molecules essentially give no signal. So if someone of you is familiar with other old, rather conventional methods to map antibody epitopes using synthetic peptides, we realize immediately that there is an important difference. It's completely it's a considerable gain in both sensitivity and specificity as compared to free-floating peptides or peptides immobilized on something different, something that is not a glass chip like the PEPperPRINT. Now, this was the first approach, and as you can see, we identified essentially the same 6-mer peptide as the target epitope of our antibody. But even more interestingly, when we ran a substitution scan of the putative epitope, we made several observations that are of considerable interest to us. Well, first of all, this is the epitope. Here you can read it on the bottom, that has been selected by scanning the entire molecule. So we'll focus on this, and each position was exchanged. So at each position we modify the epitope with all the 20 natural amino acids. This, first of all, confirms the epitope boundaries, and secondly, it shows that right in the middle there are crucial amino acids. These amino acids tolerate no substitution. As you can see, all the substitutions are reduce, greatly reduce, or completely abolish binding. And you can see it from the colors shown here. So the epitope was confirmed both in its sequence and in the size, in the dimensions, in the fact that it is just a short, continuous 6 amino acid epitopes. There is a crucial asparagine here, and I'd like you to pay attention to this because in the next slide I will show you that this asparagine, that is also a crucial residue, without asparagine, no binding, is only present in the single +1 HLA molecule that we had selected for epitope mapping. So let's see in the next slide. Asparagine is absent, is not present in any other variant, in any other allele from all the other HLA alleles that are available in public databases. So it's absolutely specific for the antigen of interest. So this is the first conclusion. But another thing I'd like to stress is that if you focus only on the 6 epitopes that form the epitope, 6 residues that form the epitope, there are just 3 possible variants and not 6,000. In other words, the 6,000 different alleles, once we focus on this narrow region, their complexities is drastically cut down to just 3 possible variants. And testing 4 variants, including the wild type sequence, and not 6,000 makes a huge difference. So, in fact, a very simple assay, like a Western blotting on the on the natural proteins, confirms that whatever we have found on the synthetic peptides also applies to the whole protein. So it's rather easy, as I wrote down, to make sure that the antibody is specific, using just a few variants and not all the variants that are present in nature. So I'd like to conclude about this slide, that the slide shows nicely what PEPperMap can do. So you cut down antigen complexity to an experimentally testable small data set of molecules. And so you can formally rule out cross-reactivity with unwanted antigen, even in complex and highly polymorphic protein systems, such as human leukocyte antigens. Now, before I move to the next slide, let me just add a consideration. So PEPperMap provides a straightforward method to map and scan antibody epitopes. When you run a substitution scan, you end up with a combinatorial checkerboard that reproduces at each epitope position a number of variants, not only the variants that exist in nature, but also variants that do not exist. So I can hardly think of doing this exhaustive substitution exercise by conventional methods, particularly by site-directed mutagenesis, because that involves the use of recombinant antibody molecules. It's a lot of work. It is something that you don't want to go through unless it is absolutely necessary. Now, using PEPperMap, now we see that the antibody is specific and discriminates even single substitutions. Okay? And this has been done just by resorting in the end of our essays to the natural proteins and relying on the combinatorial abilities of peptides for the first part. So that is possibly the Occam's razor that I was referring to in my introduction. Probably you have seen it in the presentation on the web. So what about conformation? Because the other part, this is specificity, but as I said, I was going to talk about conformation. So what about conformation? So we looked for the position of the epitope on a model of the HLA class 1 molecule. The epitope, as I just showed you, is short and continuous. And when you display the epitope on the 3D coordinates, this is the peptide, and here is the epitope, you realize right away that in the conformed HLA class 1 heterotrimers, the epitope is not accessible. Actually, it is buried underneath surrounding residues, and it is located in the floor of the binding group of class 1 antigens. You can barely see the crucial asparagine that I just mentioned to you peeping out here from a hole in the alpha helix. And if you rotate the molecule, you realize that the epitope right here is essentially buried underneath surrounding structures. So it follows that only HLA class 1 molecules that are unfolded in this region can express the epitope in an antibody accessible way. These native molecules will never be able to bind our antibody. In fact, this is in keeping with our results showing that the epitope is linear, and is also in agreement with a number of other biochemical assays that I will not show you in the interest of time. So, linear epitope, hidden, not available on native molecule. Just to make a simple case of a conformational epitope, let's compare the epitope that I just showed you, that is very linear, not accessible. Let's compare this epitope with a very well-known conformational epitope that was described in the 70s by PEPperPRINT, who's now at Stanford. This epitope is mapped, and you can see that it is discontinuous. Here you can see the heavy chain from the bottom. You can see the heavy chain residue 121. When I add beta-2 methylglobulin, you see that the epitope is conformational and complex because there is a residue sitting on the heavy chain and the residue sitting on beta-2 methylglobulin. So this is a typical conformational epitope, and in this case, you will not be able to map it on the version of PEPperMap that I just showed you. And please note how different is this antibody from the epitope that I just showed you. If you want to know more, I mean, I don't think it is worth the effort to go through the other antibodies that we mapped by this method and the specific conclusions that we reach. So if you want to know more, just write down the DOI that is indicated at the bottom of the slide. That will refer anyone of you who's interested to the European Journal of Immunology, and there you can read the whole story. So let's go to conclusions. And well, the first conclusion I like to make is a conclusion that is somewhat more general. So let's forget for a moment about class 1 major histocompatibility complex HLA molecules. Let's talk in general in terms of how proteins fold. So one question that I'm sure many of you might ask is, how is it possible that an antibody like the one I just described, an antibody to a linear epitope that is hidden in conformed class 1 molecules, how is it possible that people use this antibody under the assumption that it binds conformed class 1 HLA molecules? I mean, how is it possible that the nature of the antibody epitope is so deceiving? So I do not have a conclusive answer, but I may just note that this happens quite often, unfortunately. Why? Well, proteins are very difficult. Proteins acquire several conformations. So the idea that the protein is always conformed the same way, it is an old idea. And there are more recent models that envisage progressive maturation, folding of proteins that fall down through a free energy funnel and get trapped on ridges in this funnel until they reach a native state. and the process goes back and forth. So it goes both directions. So there is an equilibrium between folded intermediates, partially folded with heavy chains in this case, but proteins that are in a native state. And this implies that partially folded molecules do exist in nature, and actually many authors have ascribed specific functions to these partial folds that are not carried out by the native state, and vice versa. So partial folding is a very difficult state to define. And the interesting thing about these partial folds is that quite often they accumulate, and in the case of class 1 MHC molecules, they accumulate, they escape quality control by the cell and go to the surface. So if I use an antibody, I do not know the epitope, I do not know the conformation of the molecule where the epitope is carried. I mean, if I just use this antibody in the belief that it binds native molecules and I get surface binding, it does not mean just by the fact that the protein is expressed at its physiological location that the protein is folded. It may be partially folded and sometimes even deeply unfolded. And in our specific case, the epitope that we have detected is not carried by the heterotrimer, but is carried by heavy chains that have dissociated from beta-2 and peptide. The epitope is up there, and that's where the antibody binds. So, in summary, you cannot extrapolate results or even just binding studies for this molecule here to the whole pool of class 1 molecules expressed on the surface, and even less to conformed molecules that are anyway not available for binding with this specific antibody that binds a linear epitope. So if you use this antibody here with the idea that you are getting the good, the well-conformed molecules, and then you draw conclusions, and then you assign prognosis to patients with viral infections and cancer, you may do that with insufficient accuracy. One final comment. We live in a world that is dominated by committees. Committees decide minimum quality standards in scientific communication, in scientific validation, minimum requirements to publish papers and to provide information to the scientific community. But although this is quite, you know, everyone agrees that this is a very important activity, the activity of setting minimum quality standards. To my knowledge, there are few sites and repositories that pay attention to the assignment of antibody epitopes. Take most commercial vendors. They commercialize antibodies and do not bother indicating the epitope on the information sheet. I think this is quite unfortunate because without epitope mapping is not an option. I mean, without epitope mapping, you may misuse the antibodies. And this is really a pity because, in my opinion, even in the post-genomic era, antibodies remain possibly the most accurate, immediate, they are dependable, the most informative tool that we have to classify and cure. Therefore, my suggestion, my idea, the caveat that I would like you to bring home after the seminar is do not settle unless you are sure that the antibody that you are using has an assigned epitope. do not run the risk to use this antibody off-label. Maybe you think it's a native protein, maybe it's not. And I mean, it's not difficult right now. In 2015, there are many methods to map antibody epitopes. And the proteomics of epitope mapping are rapidly evolving. There are several mapping strategies. I showed you PEPperMap. I think PEPperMap is one of the best systems. These last generation glass chips are really a leap forward as compared to other solutions. They can save you time, they can save you money. Another little suggestion that I can give you is that I just read, I mean, Carsten just sent me the advertisement for the next webinar, and there are there is a new peptide mapping system commercialized by PEPperMap that makes use of constrained peptide mapping systems. And this may give you information on some, possibly not all, but some conformational evidence. I think this is a topic of considerable interest, and I will attend the next webinar by PEPperMap. Obviously, in this case, I will not talk, but I will be with you in the audience. So I'm pretty much finished. Thank you for having been with me and with my group. You can see all the people who collaborated with me in this mapping project. It's been a short critical overview of what an antibody can do for you and what you can do to get the best out of your antibody. And now I will be glad to take questions. Thank you very much.